Good evening, ladies and gentlemen, and welcome to Battling and Beating Cancer here on CAN TV. We are originating, as we always do, from our CAN TV studios in Chicago and streaming live worldwide at www.chicagobloodcancer.org and cantv.org. And we have another fabulous show for you tonight. I'm Scott Seaman, and along with Charlene McMann, who's not here today, we are the co-founders of Chicago Blood Cancer Foundation, a nonprofit committed to curing lymphoma, leukemia, and myeloma. And we really use all the tools available to us to help people who are battling the disease. And those tools include education, awareness, supporting cutting-edge research, which is going to be the focus of tonight's show, public policy advocacy, patient advocacy, and dealing with issues that are of interest to people, individuals, families who are battling blood cancer. You can reach us at chicagobloodcancer.org or by calling 888-792-9992. And if you are impacted or know somebody who's impacted by blood cancer, reach out to us and we'll help you in any way that we can. So a couple of weeks ago, Charlene was interviewing Dr. Janet Rowley, and her legacy is phenomenal, and her work is continuing. And we talked then, as we do so often, about blood cancer research being the superhighway to curing cancer, because so many of the breakthroughs and the treatments for all sorts of cancers and other diseases originated from the blood cancers, lymphoma and leukemia research in particular. And that is also the case that superhighway runs right here through Chicago, because Chicago and its great doctors and institutions are responsible for so many magnificent breakthroughs that have saved lives all over the world. And so we ask everybody to support our great institutions right here in Chicago. And we follow up on that theme tonight because we are joined by Dr. Shad Thaxton, and he is responsible for really another exciting breakthrough, along with Dr. Leo Gordon, who hopefully will be able to join us. He was treating patients up until a few minutes ago, which, believe it or not, is almost as important as joining us here on Battling and Beating Cancer. So hopefully he'll join us. But in either event, what we want to do is talk about the groundbreaking research that Dr. Gordon and Dr. Thaxton have come up with and what its great potential is. So let me just introduce Dr. Thaxton. He is a highly regarded urologist, assistant professor of urology at the Feinberg School of Medicine, and he's on staff at Northwestern at the Robert H. Lurie Comprehensive Cancer Center of Northwestern University. That's a long name. And he's really an award-winning researcher. Doctor, thank you so much for joining us, and thank you for all the magnificent work that you do.
It's a pleasure to be here. Thank you, Scott.
And so you are winning, all of the love and affection from the blood cancer patient community because of this nanoparticle technology and your studies that we're going to talk about shortly. And you didn't set out to do any of that, but it sort of happened the way so many great things happen, by doing some good work with some good ideas, and sometimes it takes you in directions you don't necessarily envision at the beginning.
Correct.
So let's start at really the basics, because we say nanoparticles, and that's a big word, and most people, dummies like me, don't even know what that is. So can you just start at the basics and tell us what nanotechnology is and what nanoparticles are?
Sure. So nanotechnology is broadly defined as the field of science that deals with materials that exist on a nanometer length scale. And so strictly defined, it is the science of materials that exists between 1 to 100 nanometers. They have a size dimension between 1 and 100 nanometers.
And so, how big is a nanometer?
Yeah, so just to give you a feel for the size of a nanometer. So a human hair is about a millimeter or a couple of millimeters in diameter. And so if you think about a human hair and you think about going about 1,000 times smaller than the diameter of a human hair, you end up with materials that are about 1 micrometer or 1 millionth of a meter in size. And so you have to go 1,000-fold smaller than that even to get down to the nanometer scale, or the size of things that are on the nanometer, or 1 billionth of a meter in scale. So to put it in biology terms, a human cell, for instance, is a good estimate for the size of a human cell. It's about 10 microns, or 10 1/millionths of a meter in diameter. And the nanometer then, of course, is 1,000-fold smaller than that. So all of the proteins and nucleic acids, really the machinery of human cells, these are naturally occurring proteins and nucleic acids that exist on the nanometer length scale. So again, a billionth of a meter in diameter or in size. And specifically in nanotechnology labs, what we focus on is actually synthesizing materials, directing the synthesis of materials like the ones that Dr. Gordon and I have used in these studies, directing them and building them from the bottom up. So starting with materials, gold, lipids, proteins, and building something useful, ultimately in this case to develop and to develop as a therapeutic for lymphoma.
And so we're talking about technology dealing with stuff that is extremely small, that up until recent times was at too small of a level for us to really address. And the opportunities presented by nanotechnology are just tremendous.
Absolutely.
Now, for your particular research, you used gold nanoparticles. And I know gold's a good thing, and all you ladies out there, but why gold as opposed to silver or diamonds, other things that people like?
Absolutely. So gold has a lot of reasons for choosing gold. The first one is that it has a track record for being biocompatible. It has a good track record for being compatible with biological systems. It's been used in humans before as a therapy.
Gold fillings.
Gold fillings for teeth, an excellent example for sure. So we start with a good foundation for a material that has a good track record for being biocompatible and safe for human use. That said, that's not to say that the materials that Dr. Gordon and I have developed certainly won't go through a rigorous evaluation process with regard to toxicity and safety in humans, but at least we're starting with a foundation that has a good track record. Other things about gold that are very useful is that gold nanoparticles, these particles we're using to develop these therapies, can be made with very discrete sizes. So what we're trying to do, and I'm sure we'll talk about it as we go forward, what we're trying to do is develop materials that look a lot like natural nanoparticle counterparts. And so when we start with a particle, what we need to have is a particle that we can make with very discrete size. We want to make and use a particle that has a 5 nanometer core, for instance. So the particles we use have a 5 nanometer core. And we want them to be 5 nanometers because we want to make something ultimately that's about 10 nanometers in diameter. So we don't want to start with particles that are 5 or 7 or 10 or a mixture of those. We want to start with 5 nanometer particles, and with gold we can make those. And that chemistry is very well established. And so we can start with materials that have a discrete size to make ultimately the particles that Dr. Gordon and I are working with. Other things, they're easy to track in the human body, which is very useful. And in other biological systems, not in the human body quite yet, but in other biological systems, gold is very easy to track. And it's relatively easy to do chemistry on the surface of the particles, for instance, to assemble the fats and the protein that we've done here to develop this nanoparticle.
And so when you started your research, if I understand this, you were actually looking for something that would help with heart disease. Is that correct?
Correct.
So
Correct.
Tell us how you started out and what you were looking for.
Sure. So my laboratory focuses on biomimicry, and biomimicry is simply a way of saying that we take pages out of nature's playbook, and what we then try to do is synthesize in the laboratory materials that look a lot like them, look a lot like natural nanostructures. And so where we started was we took high-density lipoproteins. And so high-density lipoproteins are natural nanoparticles that float around in the bloodstream, and their main function, or one of their main functions, is to bind cholesterol. And it turns out in cardiovascular disease that the higher one's level of good cholesterol carried in the context of high-density lipoproteins, the lower their risk of developing heart disease, heart attacks, etc. So we set about in the laboratory to actually synthesize these HDLs. If they're so good, then why couldn't we make a synthetic version of them to administer as a potential therapy for atherosclerosis, cardiovascular disease, and heart disease? So that's what we did. It turns out these particles are about 13 nanometers in diameter, the natural ones. So we took them and we basically made them a synthetic target. We wanted to make, from the bottom up, start with a 5 nanometer particle, 5 nanometer gold particle, and assemble on their surface the exact same chemicals, fats, and proteins that are on the surface of natural HDL. And once having those particles, the question was, do they then behave biologically like their natural counterparts? And so that's how we got started.
And before, we're going to focus in a moment on lymphoma, but how did your studies pan out in terms of helping people with heart disease?
So they're still ongoing. We have as I mentioned before, we've synthesized these nanostructures, of course, characterized them, and it turns out they are a very close mimic of their natural counterparts. And it turns out that they function pretty good in sequestering cholesterol. They bind cholesterol to their surface. And so we have a huge amount of research going on in cardiovascular disease, looking to see whether or not the particles will do as we initially sought out to do, activate cholesterol transport in a beneficial way in the context of heart disease. Those studies are still ongoing. And one of the reasons they're still ongoing and haven't progressed to the extent that the lymphoma research has is because the lymphoma research has been so exciting. We have, the lab is split, if you will, into different factions, different people working on different things. So as I mentioned, the cardiovascular disease work continues and is very exciting. We're very excited about it. But then
the lymphoma stuff on the fast track because it's really exciting.
Right, and it's progressing relatively quickly.
So as I understand, as fate would have it, Dr. Gordon was listening to a lecture that you were giving, which is a little bit unusual because usually Dr. Gordon is giving the lectures because he is one of the most prominent oncologists and lymphoma doctors in the world. Mm-hmm. But he was in the audience listening, and then a light bulb went off, I think, in his mind because he had observed with patients with B-cell lymphomas would have lower levels of HDL cholesterol.
That's correct.
That right?
That's correct.
And so you two hooked up.
Right. Exactly. So, as the story goes, I was giving a seminar on the synthesis and the characterization of these materials, their ability to bind cholesterol, traffic cholesterol. And another thing that I mentioned during that talk was the fact that these particles may in fact be very good vehicles to deliver therapy, to deliver drugs, right, using the particles. And so, as you mentioned, because of Dr. Gordon's knowledge about HDL biology, lipid biology in patients with lymphoma, and because of his interest in developing new ways to deliver other chemotherapies to lymphoma cells specifically, we initiated a conversation that led us down this path of using the high-density lipoprotein mimic potentially as a drug delivery vehicle, and as we later found, as a standalone therapy, a potential standalone therapy for lymphoma.
So Dr. Gordon's focus was if we could use these nanoparticles to transport or get other drugs where we want them, whether it's chemotherapies or other chemicals, that would be a marvelous thing because it would be a new vehicle to convey them.
Correct.
But you guys found that you got a lot more than what you bargained for.
That's right. So in our initial experiments, we did two things. We took the particle, the vehicle alone, if you will, just the nanoparticle, and then we took another set of nanoparticles where we had added a drug to those particles. We then exposed lymphoma cells to these particles, the two different particle types, and lo and behold, the particle by itself, the HDL nanoparticle by itself, performed just as well in killing lymphoma cells as the particles did that had the drug added. And so,
what drug was added?
It was a small molecule chemotherapeutic that we had put on the particle, and the hope was that the particle would then deliver specifically to the lymphoma cells, looking far down the line versus normal healthy cells. And as I mentioned, we saw that the therapeutic effect with the particles by themselves. And of course, the first thing we thought was that we'd made a mistake and we'd switched them, or something had contaminated the solution, and it wasn't a real result.
And we've seen this before, where for example, with Rituxan, monoclonal antibody, we knew it was working, but we thought it was working because it was killing the cancer cells directly. But once we understood it better, we understood that that's not what was happening. It was attaching itself to the protein of the cells and harnessing an immune reaction so that a person's immune system would fight off the lymphoma. Mm-hmm. And so here you're going for another vehicle. Mm-hmm. Instead of delivering radioimmune therapy, as in the case of Rituxan plus a radioactive isotope, you've got something, you're delivering a chemotherapy agent, but you're discovering that another batch of the stuff is doing the same thing by itself without any chemical design or what you thought was designed to do it.
That's right.
And so right away you've got to be excited because you could be famous, you could have a drug named after you, you could have something that could really save hundreds of thousands of lives.
Most importantly that. And, so we were certainly...
Although the name thing's good.
as we interpreted those results and we moved forward. Of course, the question became, it was a reproducible one, of course. So we were able to reproduce multiple times the fact that this drug, at least in cultured human cells, in the petri dish, if you will, was effective. And so the next question for Dr. Gordon and I really became, so how is this thing working? Exactly what is the particle binding and latching onto? For instance, you mentioned with Rituxan, there's a specific protein on the surface of the cells that this Rituxan binds to. And so our question was similar. How are the particles actually functioning to kill lymphoma cells? And that became kind of the next focus of our work, is to better understand that.
And so you've actually got some degree of understanding, because it turns out you guys aren't only fabulous researchers, but you're spies and you're double spies, and you work some torture techniques such as starvation, because these nanoparticles got in, they fool the cells. The cells thrive on cholesterol. They think they're eating cholesterol, but they're eating these nanoparticles that are blocking them and killing them off. It's probably not the scientific way to say it, but is that? That's
basically it, and that's a perfect way to say it. And it goes back to originally what we were trying to do. So we were originally trying to develop nanoparticle mimics of their natural counterparts. And so what we hoped that they would do functionally was to pull cholesterol out of cells. And it was interesting at the time. It wasn't something that we fully appreciated at the time, and we certainly appreciate more now, was this notion that the particles are a lot like natural HDLs with regard to their surface coating. But there's one, of course, critical key difference, and that's in natural HDLs, there's a very cholesterol-rich core. And of course, in the mimics, in the particles that we've made, there's a gold nanoparticle in the center of ultimately the construct. And so it turns out lymphoma cells latch on. They have surface proteins on the surface of their cells that bind HDL, and then they pull the cholesterol from the particle into the cell. And it turns out that they're very dependent upon that cholesterol meal, if you will, for survival and growth. On the other hand, when the nanoparticle binds to the same receptor, same proteins on the surface of those cells, and then it does two things. Number one, it prevents natural HDL from binding and delivering that cholesterol-rich meal. And number two, it's actively starving the cell of cholesterol by pulling it out and then not delivering anything from the core of the particle, which of course is occupied by gold. So it's kind of this, people have described it as, the cell bites down on what it thinks is a juicy cholesterol meal, and of course gets a mouthful of gold and not a mouthful of cholesterol. And it's that type of mimicry and receptor binding that we're able to take advantage of, interestingly enough, to starve these cells of cholesterol and ultimately kill them.
Well, and of course, on Battling and Beating Cancer, we love life and we love people, but we hate cancer. So anything that kills those cancer cells is terrific. And so how far have you gotten in terms of figuring out whether or not this type of therapy could work for all types of B cells or some types of B cells? And is it only seem to work for B cells? So far, we focused on diffuse large B-cell lymphoma. And so a recently published paper by Dr. Gordon and myself is really focused on diffuse large B-cell lymphoma. We do know that the receptor, this protein that's on the surface of the cells that the particles are binding to, is expressed by multiple different cell types. And we're just now beginning, as we understand the mechanism a little bit more of how the particle works, in addition to the cholesterol story, beginning to look at other of these cancer types, including lymphomas, leukemias, etc., and all types of others, breast cancer, prostate cancer, etc., to see whether or not they're more broadly applicable. And so it's going to be a process of trial and error. And since you're already dealing with one B-cell type of lymphoma, which is a good one to deal with. We've talked many times here about follicular lymphoma, but things like mantle cell lymphoma, large diffuse B-cell, which was a type I had, although there are many good treatments for that, is a logical place to start. Now, has any of this testing been done on humans?
None of this testing has been done on humans, but all of the cell lines, of course, that we use, these are immortalized cell lines derived from human patients. And so that's where we typically start. We've also studied these in mouse models of lymphoma, and so there it gives you just a better idea that administration of the materials, the administration of the particles into the bloodstream in a mouse model of lymphoma works. And so we've done those studies, we've published those studies showing that at least in the mouse model we get a nice tumor response and an inhibition of tumor growth as we deliver the nanoparticles.
So you start out the cell lines.
Mm-hmm.
You've got some animal study confirmation.
Mm-hmm.
I don't know whether or not you use rats, which are sort of like the animal equivalent of lawyers, very similar. And so if it's worked on rats, it could work on some humans.
Okay.
But then you've got to go down that whole phase of clinical trials and things before you even get to clinical trials.
Correct. Exactly.
And we'll talk about that because I think it's important for people to understand how many steps it takes to go through from having a magnificent idea until it actually translates into therapies that get themselves employed in patients.
Mm-hmm.
But the great findings so far suggest that we've got an effective treatment,
Mm-hmm.
at least for follicular lymphomas. We've got something that looks like it may be effective for other types. But the other encouraging aspect is there doesn't seem to be much, if any, toxicity associated with this treatment. Is that right?
Correct. Yeah, that's another big part of our work is, in addition, of course, yet understanding that it works, understanding that it's not harming normal cells. And so we've done a lot of those studies. We've looked at a lot of normal cells derived from humans to make sure that there is not toxicity to those cell types. Again, we did the animal model, the mouse model, and demonstrated that there was no obvious toxicity in that context. But of course, we're still at the beginning along this road to go all the way through to an application for an investigational new drug with the FDA. There needs to be further scrutiny, not only on the particles themselves, but also on their toxicity or any potential toxicity that they would have. All of that testing needs to be done. And then ultimately approved by the FDA, we hope, as an investigational drug. So at that point, one could start an early phase 1 trial in humans.
So it is a long road. I mean, we could be 3, 4, 5 years or more away from the point where it gets to clinical trials that patients in a clinical setting are at least able to get it there, even if it's not generally available to patients outside of clinical trials.
Correct.
And just tell us briefly what your next steps are, where you're going, and how you plan on getting there.
Yeah. Really the next steps are to evaluate, further, do a lot of the studies that we've mentioned, further evaluate the mechanism of action, while at the same time repeating our studies in animal models, clearly evaluating the toxicity of the particles, and ultimately, we hope, in the next 18 months to a couple of years, get these particles through and into phase 1 trials.
But this may be the next Rituxan, the next Gleevec. I mean, these are how these miracles happen, not all at one time, but a lot of good scientists doing a lot of good work, jumping through a lot of good hoops, sometimes taking some twists and turns that you don't expect. But all of this, of course, takes money. And we're living in an environment now where for several years now the real rate of the government's investment in research has been declining. The last year or two, the nominal investment in research, and this is before some of the budgetary issues we're dealing with now. So one of the things that people like you and lots of good researchers working on other things need is money and private philanthropy and donations. Is that
correct? And we've brought this this new nanoparticle treatment for lymphoma up to a point where people often call it the valley of death, where a lot of really good ideas and a lot of promising technologies come and ultimately come to rest unless there's this influx of interest and money ultimately to drive them across this valley of death and ultimately into patients where they can hopefully have the biggest benefit. And so, that becomes ultimately an issue that we grapple with as scientists. We certainly appeal to, as you mentioned, philanthropic groups, private individuals, the United States government, of course. They help and they move these therapies along. So we're certainly hoping to get support from all of these different entities to move this therapy into human trials.
Well, Dr. Thaxton, thank you so much. This is so exciting. Once we did an article on this, and we led the word out that we were going to have our show on this, and we had all sorts of people emailing and Facebook chatting because it's really exciting to a lot of patients. Of course, there's going to be a little time and a little investment yet to do, but because of good work by good doctors, all of us who have blood cancer and other types of cancer really have every reason to be hopeful. And now more than ever, because of the good that the money does, because we're at that point now where because of the understanding of microbiology that we have, because of the Genome Project, it really is paying off in terms of timing and in terms of extent more rapidly than ever in the past. So when all this federal funding is shrinking for cancer research, it's a time when cancer research investment would do the most good. So Dr. Thaxton, thank you so much for joining us, and we are going to continue to bring you more of the important, informative shows, keep bringing the best doctors and researchers right into your living room, so that you know when you're dealing with cancer that there are lots of resources, lots of good clinicians, lots of good researchers, and organizations like Chicago Blood Cancer Foundation that are there to help you, because when you're dealing with the diagnosis of cancer, and 1 out of 2 men in America and more than 1 out of 3 women will hear that phrase, "you have cancer," during their lifetime, it's important to know that there's every reason to be hopeful, and that hope comes from research, because research really is the key to all of our long-term survival. Now, you can see this program, all of the prior programs from this season and prior seasons, at chicagobloodcancer.org. You can go to Chicago Blood Cancer Foundation's YouTube channel, and we download them there. Because some of these programs may not be meaningful to you now, but when you or someone you love is diagnosed with a particular type of cancer or blood cancer, you can go back, refer to that information, go to chicagobloodcancer.org. We have lots of other information about lymphoma, leukemia, myeloma, the latest breakthroughs coming up this summer. We're hoping to have a new page up that's going to chronicle all of the clinical trials going on in blood cancer here in Chicago. So, we thank for Charlene, our beautiful, wonderful executive producers. Would like to get them in front of the camera, but they say no, they've already had too many marriage proposals. They don't want to increase that. So our thanks to Dorothy, to Christy, to our CAN TV engineering crew, and most importantly, our thanks to you. We thank Dr. Gordon, who was unable to make it tonight, Dr. Thaxton, who was, for their marvelous research. And you've seen, and you can go look at some of our videos from past shows and see Dr. Gordon talk about different types of lymphoma, including follicular lymphoma. So we want you all to join us on the superhighway to curing cancer. So remember to keep a song in your heart, and always keep some heart in your song. On behalf of Charlene, we thank you for joining us. Good night. Good health, everybody.